Leading a startup team? Whether you're delivering a sugar rush, stocking coffee, or getting a regular delivery of snacks, Office Depot has solutions that fit every startup culture, from getting those first business cards and stationery to ordering fleece pullovers with your new logo. To learn how Office Depot and the California Technology Council have partnered to bring you savings on all of these startup essentials and more, go to californiatechnology.org/member benefits. I'm Daniel Levine, and this is the Bio Report. Cidara Therapeutics is developing new anti-infectives using its Cloudbreak platform to develop antiviral conjugates that both directly kill pathogens and also direct a patient's immune cells to attack and eliminate them. The company is advancing its first therapy developed through the platform to the clinic, CB012, an experimental therapy that's being developed to both treat and prevent flu infections. We spoke to Jeff Stein, CEO of Cidara, about the therapy, how it works, and the need the company is trying to address. Jeff, thanks for joining us.
Absolutely. Pleasure to speak with you again.
We're going to talk about influenza, Cidara, and your efforts to develop an experimental therapy as both a treatment and a prophylactic against flu. Perhaps we can start with the opportunity. I think most people dismiss flu as a fairly benign seasonal illness, but from a public health point of view, it does have significant consequences. How big a problem does it represent?
Well, it's an enormous problem when you consider that last year some 80,000 people in the U.S. died of influenza. 650,000 people globally died of influenza. It is an enormous problem. The CDC recommends that every individual in the U.S. over the age of 6 months should be vaccinated for influenza. Unfortunately, fewer than 40% get vaccinated, and even those that do get vaccinated, up to around 40% are responding to the vaccine. So what that means is that around 250 million people in the U.S. alone are vulnerable. They are unprotected against influenza. So that's a big number. So this year, the number is running a little bit smaller, probably tracking towards about 60,000 deaths in the U.S., but that is a very big problem, and especially what we call the vulnerable population, meaning the young, the elderly, and those that are immune compromised, or the vulnerable patients, which comprise about 25% of the U.S. population.
Every year there's a new flu vaccine. How effective are these generally?
Not very effective. For a couple of reasons. Number one is the long manufacturing process. Every year, the CDC is in Asia. Your virus hunters are there trying to make predictions of which influenza strains are going to be a problem in the U.S. when the flu season hits. So they're making a guess, and then you have this relatively cumbersome and antiquated system of growing the virus and generating the antigens in chicken eggs. And that's a 6-month process. And during that 6-month manufacturing process, that influenza virus can mutate. It can change so that it can avoid the vaccines. And consequently, it's, the vaccines on an annual basis are effective in about 25 to 50% of the people, averaging around 40% of the individuals who get the vaccine. And this most current year, even though it is milder influenza strain than the previous year, the season is longer. And so, as it turns out, to be fully protected for the flu, for the entire flu season, it looks like you need to have 2 vaccinations. And so that's really a problem because most people find it a challenge to get even a single vaccination.
There are drugs available to treat flu. These have to be administered soon after onset of symptoms to be effective. There are neuraminidase inhibitors, and last year the FDA approved an endonuclease inhibitor. How do these drugs work, and how good a job do they do?
So Tamiflu is the neuraminidase inhibitor that is most prevalently in use, and the newer drug, the endonuclease inhibitor, is a drug called Xofluza, which is being marketed by Roche Pharmaceuticals. Both of these drugs can work if administered shortly after the appearance of flu symptoms. In general, they have to be administered within 24 to 48 hours of the first appearance of the flu. And that can be a challenge because, I think everyone who has experienced the flu knows that you don't really know what it is for the first 24 hours. And then when it hits, you have to get yourself to a doctor. And most people who receive that drug or those drugs get it outside of that window where they no longer can be very effective.
When you were last on the show, you discussed Cidara's anti-infective immunotherapy platform, Cloudbreak. Can you remind people what it is and how it works?
Sure. I think it's easier to describe what it is not. So it is not a traditional vaccine. It is not a antiviral drug per se like Xofluza or Tamiflu. And it's not a monoclonal antibody. So you've got to keep that in mind because that's probably the biggest challenge in describing this program. We call it an AVC for an antiviral conjugate. So it's a bispecific, meaning there are 2 parts to the molecule. The front end of the molecule is a novel antiviral drug that binds to and can independently rapidly kill the virus. The back end is a fragment of a human antibody called the Fc domain, and that engages an immune response. And so you get a combined effect of a very potent, fast-acting antiviral drug combined with a local amplification of the immune system right on the virus. And that combination is extremely potent. And importantly, this drug can be administered potentially once the flu season. We have to demonstrate that in the clinic, obviously. But based on the data we have to date in animal models, we believe that this drug can be administered as a small subcutaneous injection once the flu season, so it can be administered as a prophylactic, especially in vulnerable patients, or it could be administered directly as a very potent, fast-acting drug with the potential to expand that treatment window beyond the 24 to 48 hours that is the limitation of currently available antivirals.
Is the expectation is that this would have potential against all strains of flu?
Yeah, that's an important distinction. The vaccines that are developed on an annual basis for seasonal flu are generally active only against influenza A, have no activity against influenza B strains. And the same thing with the currently available drugs. And because of that limitation, the current activity in influenza research has been on combining an antiviral with, say, a monoclonal antibody against influenza B in order to get broader coverage. Our AVC molecules don't have to do that because they're actually a combination in the single molecule. And they're broadly active against influenza A and influenza B, basically all the seasonal as well as the pandemic strains of influenza.
As a treatment, does its efficacy depend on how soon after symptoms arise it's administered, or can this work at any time?
Well, based on the animal data that we have to date, it suggests that we can expand that treatment window from that 24 to 48 hour limit that the existing antivirals have up to 72 hours. So that's an important expansion of the current treatment window, and it should enable more patients to be effectively treated.
Since it has this ability to confer long-acting protection against infection, could it be used as an alternative to vaccines? And if so, would it represent some sort of pan-flu vaccine?
Well, we believe that it certainly could be administered like that. We would test it on top of existing vaccines in the clinic and offer it as something that could bolster the efficacy of existing vaccines. We certainly don't want to recommend that people don't get a vaccine, because that is by far the cheapest and fastest way to get protected against the flu, despite the limitations of current vaccines. We would suggest that this is something that in the vulnerable patient population, the young, the elderly, the immune compromised, that it would make a lot of sense to put this on top of a vaccine.
And is that the long-term strategy, given how relatively inexpensive flu vaccines are, that this would be targeted at specific subpopulations for use?
That's right. If you speak with physicians who treat hospitalized patients, they simply do not have any alternatives. They know that if you have a debilitated immune system, such as those vulnerable patient populations I mentioned, you don't respond to vaccines. You need an active, robust immune system in order for vaccines to be effective. And similarly, you have these limitations of the existing drugs. And so this is where you get the highest mortality rate, is in these vulnerable patient populations, and they simply have no tools to address that.
You, I believe, indicated this is an injectable drug. How is it administered, and how often does someone have to be exposed to it?
Well, from our animal testing, it appears that we can get very rapid exposure of the drug systemically by either IV, intramuscular, or subcutaneous injection. Subcutaneous being very similar to how a flu vaccine is currently being administered. It is not orally available. It's a large molecule, but we get very rapid exposure by the other three routes of administration. So it's very similar to what people would be accustomed to in getting a flu vaccine. And with respect to the frequency, again, our goal is to have this be effective once per flu season.
And what do we know about the experimental drug from preclinical tests to date, as far as its safety and efficacy?
Well, it's a really important question. Those of us who have been involved in the program have never experienced a discovery and development program like this. Typically, when you're in this stage of development, you bump up against certain limitations of efficacy, safety, or pharmacokinetics, which drives you in a certain direction. For example, it's not effective against influenza B, so we're going to focus on influenza A. It doesn't have the pharmacokinetics that will enable once a flu season, so it's going to be administered daily, and so forth. In this program, we haven't run into any of those limitations. So up to the maximum dose tested, we have seen no signs of toxicity. At the lowest doses tested, we see very robust potency. And at single doses, this drug provides very long-term protection regardless of the route of administration. So perhaps the biggest challenge in the future clinical development is, where do we take it? We've had some of the key opinion leaders in the U.S. Review the data, and we pretty much get kind of a split opinion depending on the area of expertise. So you have a hospitalist who say, well, you have to develop this as a treatment in my vulnerable patients. And you have the vaccine experts who say, well, no, this has to be developed as a prophylactic. So it's a very high-grade challenge to have. But we're involving some of the leading experts in the U.S. and in fact, globally to help guide us through that clinical development process.
Well, what does the potential dual usage of the drug say about the clinical development path forward?
Well, probably the strongest opinion and the biggest unmet need is for prophylaxis in the vulnerable patient population. Those three cohorts that I mentioned, the young, the elderly, and the immune compromised. And that latter category of patients is growing. These are patients with autoimmune disease. You're seeing a proliferation of novel autoimmune drugs, and the way those work is that they suppress the immune system. And any individual who has a suppressed immune system are less responsive to vaccines and are highly vulnerable to influenza. And when they get influenza, it tends to be much more severe. So prophylaxis in that focused patient population, which, I say focus, it still represents about 1/4 of the U.S. population, is probably a direction that we will seriously evaluate once we get into clinical testing.
If all goes well, when might you have the first readout of data from trials in human subjects?
Well, the challenge with this type of molecule being completely new is in the manufacturing. So we would expect to file IND, Institutional New Drug Application, to begin phase 1 testing at the end of next year.
Jeff Stein, CEO of Cidara Therapeutics. Jeff, thanks so much for your time today.
Danny, thank you very much. It was a pleasure.
Thanks for listening. The Bio Report is a production of the Levine Media Group. To automatically download this podcast each week, subscribe to our RSS feed or through iTunes or other podcast manager. To join our mailing list, go to levinemediagroup.com. We'd love to hear from you. If you want to drop us a line or are interested in sponsoring this podcast, send an email to danny@levinemediagroup.com. Special thanks to Jonah Levine, who composed our theme music, and the Jonah Levine Collective, which performs it.